Welcome to Exam Mini Video 5. Today we are going to be talking about DNA mutations, specifically different types of point mutations. Before we begin, here are some terms you should know to better understand this topic. Please pause if anything seems unfamiliar. Looking at this section of double-stranded DNA, we can see that the top strand is the template strand and the bottom strand is the coding strand. For the rest of this video, we will only be using the coding strand, so let's flip it so that from left to right it is in the 5' to 3' orientation. The coding strand gives us the same coding sequence we will see in the transcribed mRNA, except that all the thymines will be replaced by uracils, giving us this mRNA sequence. We now need to identify the start codon AUG, which is here, to determine where translation will begin. Now we must divide the sequence that follows into reading frames to determine the codons that will dictate the translation of this sequence of amino acids. The translation of this sequence is analogous to a train running on a track in which the pieces of the track are the amino acids and the train serves as the functional characteristics of the produced protein. In the case of a sequence like this that lacks mutations, the train runs smoothly and slows down to stop and arrive safely at the train station. But what happens when there are mutations? One type of point mutation is a silent mutation in which the substituted nucleotide has no effect on what amino acid is coded for. So if we had a mutation here and a transition occurred replacing a cytosine with a thymine, the mutation would trickle down through transcription, changing our second codon. However, this changed codon still codes for valine, so the produced protein does not change, and our train still runs smoothly and arrives safely at the station. The second type of point mutation is the missense mutation, in which the substituted nucleotide changes the codon so that it encodes for a different amino acid. So if we had a mutation here, and a transversion occurred replacing a guanine with a cytosine, the mutation would trickle down through transcription, changing our third codon. And this time, the changed codon codes for serine instead of for arginine. In this case, the length of the produced protein has not changed, but the amino acid sequence has. So while the train may run smoothly, it arrives at a different type of station producing a different protein. The third type of point mutation is a nonsense mutation, in which the substituted nucleotide changes the codon so that it becomes a stop codon. So if we had a mutation here, and a transversion occurred replacing an adenine with a thymine, the mutation again would trickle down through transcription, changing our fifth codon. And this time, lysine is replaced by a premature stop codon, so that when the train moves along the track, the protein is cut short, or the train does not have enough time to slow down, and the functionality of the protein is damaged. The final type of point mutation is a readthrough mutation, in which the substituted nucleotide changes the stop codon so that it encodes for an amino acid. So if we had a mutation here, and a transversion occurred replacing a thymine with an adenine, the mutation would once more trickle down through transcription, changing our sixth codon, replacing the stop codon with lysine and allowing the codons that follow to also be translated. In this case, the train does not have a signal to slow down or stop, so it barrels down the track and blows past where it should have stopped, creating an excessively long protein that may be dysfunctional. That is the end of this video. Stay tuned for another in 2 weeks.